Going through our outline, we will come to know that many of alternative form of genes we have discussed is alleles and then some inherited characters and something else. But first of all, I'm going to revise something from the previous lecture, some concepts from the previous lecture, so that we will not have to revise those again and again in the lecture in today's lecture. So first of all, we did discuss about the haploid and diploid concept, and then we came to know that when how a cell becomes a diploid and how a cell becomes a haploid. Diploid cell is always means that it is a cell which is containing two sets of similar chromosome in its nucleus, and haploid was like a single set of chromosomes, single set. The single set means minimum chromosomes required by that particular organisms. For example, if human haploid set is 23, it means that this is the minimum number of chromosomes human cell or human nucleus required to survive. These are the minimum number of chromosomes. Then I did discuss about the homologous chromosomes, that there are two sets, 23 plus 23, and every chromosome of one set is similar to the chromosomes of the other set. In this case, we had an example that if we have a single set of red, black and blue markers, and there is another set of red, black and blue markers, so black is similar to the black of other set, red is similar to to the red of other set. Similarly, the first chromosome of 23A set would be similar to the first chromosome of 23B set, so it would be called as, these would be called as the homologous chromosomes. So this was another concept. And then we said that if both of these are similar, one is similar to one, two is similar to two, it means that both have same genes, or at least same type of genes present on the same location. For example, if there is a set of that was given to the offsprings by the mother, and that was inherited to the offspring by the father, then these are two homologous chromosomes, and homologous chromosomes mean these are similar chromosomes, and similar chromosomes mean these would have almost same nature of genes. So if there is a gene at chromosome number 1, sorry, at position number 1 of this chromosome, and if it is homolog, then there would be a gene at the position number 1 of this homolog as well. Why? Because these are homologs. For example, let me continue this concept. You have two chromosomes, one is of this size and one is of this size. This is 23A set and this is 23B set, right? So this is chromosome number 1 and this is chromosome number 2. If you have if you have gene A on chromosome 1, then it would never happen that chromosome 2 would also have gene A. No, it might have the gene Z on placed on the first position. Let me repeat this. If on the first position, on the very terminal position, if the chromosome number 1 has the gene A, the chromosome number 2 would not have the gene A at the same position, because these are non-homologs. These are non-homologs. But whenever we talk about those chromosomes which are homologs, which are similar to each other, they would have the same genes on the same position, same location. Let me tell you another thing. The position of gene, I'm calling repeatedly, I'm telling you repeatedly about the position of gene. There is a very particular term of about the position of genes in the genetics. That position of the gene is known as locus, which is derived from the word location. Location mean position. So locus. And if we are talking about the location of multiple genes, so for plural we would be using the word loci, loci, loci. So if the at chromosome number 1, the first locus is containing the gene A, the chromosome number 2 would never have the gene 1 at gene A at first locus. Why? Because these are non-homologs. So this is a very critical concept, and you must be understanding it very clearly that if both are homologs, then they both would have similar nature of genes at a particular locus, at the particular loci, and these, if these are non-homologs, then they would have different genes at that locus. So this is a straightforward concept. Is it clear?
Yes, sir.
Okay, coming back to the concept about recessive, dominant, etc. I did tell you that we have been discussing since 4 or 5 days of our lectures that uh, the DNA has special segments also known as genes, and these genes would be going through a process of transcription and translation. It's especially, and the translation would be creating a protein. I did tell you that sometimes sometimes genes don't work. Sometimes genes don't work or work falsely. Very important word here, or work falsely, right? For example, I have this gene, I have this small gene here, and it is creating a protein like this, an X-shaped protein, right? So somehow this gene got mutated. I'm not discussing the concept of mutation here because it is in our curriculum, but it would be discussed later. Somehow it got mutated, it got changed, and what happened? That the shape became distorted. The shape became distorted. This is distorted shape. Now this is called as false working. This is false working. So this is a distorted protein, and this protein is useless to us. You may create hundreds and thousands of distorted proteins. If this protein is of no use to us, then we do not need that particular protein. I did mention in the previous lecture about diseases. For example, I did mention about a disease known as sickle cell anemia, and I would be discussing it again in inshallah in the coming lectures. Sickle cell anemia. There is another disease, hemophilia. Then I did discuss about the thalassemia. Thalassemia in which the hemoglobin is not formed. Hemoglobin, let me tell you a little bit about hemoglobin again. HB, it is also mentioned as HB. HB is a cluster of four proteins, four protein chains. First chain, second chain, third chain, and fourth chain. What is the function of hemoglobin? Can you tell me?
It carries oxygen.
It carries oxygen. Every chain of it contains the oxygen, one molecule of oxygen. Assume that the genes for the HB become distorted, become mutated. What would happen? They would not they they do not carry oxygen. They would never carry the oxygen. What would happen? We would be needing hemoglobin or blood from the external sources. I'm only telling you about this disease to emphasize on the importance of proper working of the gene. So coming back to the point, I did also mention in the previous lecture that if we have the two sets and I have this chromosome number 1 here, for example, this is chromosome number 1 here. If one of my gene is faulty, for example, I mention it with the small a. For example, this is the locus of gene A, and this is the locus also of gene A. But this is the form that is creating the proper protein, and this is the form of the allele. Both of these are alleles. We did mention it that the gene or the alternative form of a gene present on a homologs are alleles. We did mention it here. So if one of the alleles is working and the other is not working, it is quite fine. If both, for example, uh, this is capital A. I mentioned here capital A. Both are properly working. It means that both are creating proteins and both are working properly. But if this also becomes mutated and this also becomes mutated, what would happen? Both will not create a protein. So let me tell you very clearly one thing, that dominant, there is a concept of dominant and recessive alleles. Dominant alleles and recessive alleles. Be with me very attentively. So we must clear this concept here. Dominant alleles and recessive alleles. Dominant alleles are those which are creating a proper protein. Proper protein that is actually required by the plant. Actually required by the plant. Oh, sorry. Actually required by the organism. So that allele would be called as a dominant allele, and the other one that is not creating a proper protein, that would be called as as the recessive allele, improper protein. This is an improper protein. So uh, now we have the concept of dominant and recessive alleles. And if you talk about the inheritance, if you talk about the genetics, if you talk about the Mendelian genetics, you will come to know that there is a huge importance of dominant recessive and recessive allele in the inheritance. If one allele is dominant, it means almost 99% times, 99% of times, dominant allele means that it is creating a structure that is normal to the human or normal to the organism, that is contributing in the survival of the organism. For example, if you must have studied the structure of the eye. There are two types of cells in the eye, rods and cones. Have you studied it?
Yes, sir.
Okay, rods and cones create the proteins. Okay, so if the rods and cones are creating proteins and they contribute in our survival, normal survival, we are examining and we can understand the colors, then it is contributing in our survival. We can differentiate between the colors, so we can differentiate between an animal and an object if they are too closely, if they resemble very closely. For example, there is a very slight shade difference of color between an animal and a wood. We have the capacity to differentiate between the colors, so we can survive the attack of that animal. An example, so if a person does not have a proper protein and he or she cannot create a proper protein, he would not be able to differentiate between the two types of the cells, the two type of the colors, which means that it would be a hazardous feature for that organism. So 99% of times, dominant character is a character, or dominant allele is contributing to our survival, contributing to our survival, right? So uh, there is another form of concept about the genes and alleles. But first, tell me, are you understanding all this? Dominant and recessive alleles?
Yes, sir.
Okay. Now, talking about the alleles, we did mention here it is alternative forms of genes at the same locus of two homologs. Two homologs. For example, these are two homologs. I'm repeating many things many times. Why? Because these must be hammered. If these are not hammered, these can lead you to confusion while attempting the exam. Alternative form of genes at the same locus of two homologs. These are two homologs. And for example, this is locus number 3. This is locus number 3 of the other homolog. If this is a C gene, then at the same type of the gene would be present at the same locus of the other homolog. So about the alleles, what did we mention? That sometime one allele work and other doesn't. This is, we did discuss this few seconds earlier. And then we also mentioned that both alleles work. Both alleles work properly. And then there is another condition, both do not work. So it means that we are now I am going to tell you a proper term about these alternative forms. There is a term for these alternative forms of the gene. That term is known as genotype. That particular term is known as genotype. For example, if both the genes are same, either dominant or recessive, I'm not mentioning chromosomes here. I'm just mentioning two alphabets. If both the alleles are working properly or both are not working properly, the combination of this combination would be mentioned or would be regarded as homozygous alleles. Or you also may mention it as homozygous combinations. Homozygous combination. Homozygous combination mean that both either are working or both either are not working. Both alleles. There is another case. One is working and other is not. This combination, this genotype would be regarded as heterozygous combination. Heterozygous alleles. One is working and other is not. So genotype is the type of combination. It's the type of combination of alleles. Let me mention it, or let me write it in a proper way. If I write the genotype here again, then I would be mentioning it according, if I'm for example, if I'm writing in the exam or if I'm writing an article, I would be mentioning genotype combination of allele. First type is homozygous and the other type is heterozygous. In homozygous, both can be both can be capital or both can be dominant, because I am mentioning dominant as capital. I think you have seen here. Those which are properly
Yes.
working would be called as dominant. So it means that if it is mentioned in the capital, it means these are dominant. And if both are not working properly, these would be regarded as recessive. So I am making a genotype here AA. This genotype now completely would be called as homozygous dominant, because both are same and both are capital or both are properly working. And if I'm mentioning here homozygous recessive, it means both are same and both are recessive, both are not working. So it would be a homozygous recessive genotype. Okay. What is heterozygous? Heterozygous means both are different, a and small a, one is working and another is not working. So genotype is a very critical term using that is used in the genetics, and we are going to make the crosses according to the article of Mendel, and these terms would be widely used there while we are studying the experiments of Gregor Mendel, and I think you would be, inshallah, it would be very helpful for you. So genotype, and there is another term called phenotype: dominant, recessive, homozygous, heterozygous, genotype, and now I am going to mention a phenotype. Phenotype. What is a phenotype? Now, let me tell you very clearly. Phenotype is expressive form of a gene. Expressive form. Okay. Expressive form of a gene. Let me uh, tell you this in a very simpler way. Let me think about it. For example, let's take the example of markers. I'm a teacher, so I would be mentioning the examples of the markers. You have a marker that is apparently red. Apparently, the nib is appearing red, the lid is appearing red. But when you start writing from that marker, the ink is black. Assume that. You know, the appearance or the expression or the roughly by just looking at the marker, you said it is red.
Red.
But when you started writing, you came to know that it is black. The expression of that marker told you uh, about the quality of that marker, or you may say about the hidden quality of that marker. Let me apply this example on the human body. For example, if I have if my nucleus have the hemoglobin gene that is known as HB gene. HB gene is there. HB gene is there in the nucleus. It is present. But how would it let me realize? How would I realize that I have the HB gene? I would never realize until and unless it expresses itself. And how would it express itself? Yes, how would it?
So since hemoglobin carries oxygen, so if the oxygen levels in the body are stable and they're all right, so it means that the hemoglobin is working properly and the person has sufficient amount of it. And if it doesn't, which is the
Yes.
opposite, so then it means that the person doesn't have much hemoglobin and it's faulty.
Okay. Let us put this another way. Uh, I said that hemoglobin gene is there in my nucleus. The If the hemoglobin gene is working properly, then my level of hemoglobin is normal, which you said we can guess by the trapping of oxygen by our blood. If our blood is trapping the oxygen properly, if our blood is taking in the oxygen properly, it means we have the sufficient and recommended amount of hemoglobin, and it means the gene is working properly. So it means that we have the genes of a particular character inside our body, but the expression of that gene would be called as a phenotype. For example, and let's take another example, a very easy example. I refer repeatedly back to the blood because the example from the blood are very commonly used by our population. Non-scientific persons can understand the blood very easily. I have we especially Pakistani people, we have the blood groups of A, B, O, and there is another group, AB. And I think that Asian people, not only Pakistani people, we have these four types of blood groups. Okay, it means I have the blood group A gene. If I have the blood group A gene, then it would be creating the A blood group. So it means that I do have the gene, but that gene is expressing itself by a particular protein, and that particular protein was named by the scientists as A. So whenever a gene expresses itself, it can only express itself in the form of protein, and that expression of that gene is known as phenotype. Expression of the gene is known as phenotype. So these are the two very important terms regarding the genetics. To understand the genetics, first term is genotype and the other term is phenotype. So be very particular about this, and be very vigilant while reading or while understanding the concept, so that you may not get deceived by these terms. For example, Some students mix these terms. For example, they understand the concept of genotype as phenotype, and they applied the concept of genotype on phenotype and phenotype on genotype. They intermix these terms, never intermix these. The combination of genes is genotype, and it is appeared in the name, and phenotype is expression. So these are very important terms, and we must be very clear about this. Yes. Anything else?
Yes, sir.
Okay. Okay, Zainab, do you want to revise the what was I going to ask you? Yes, cell division or not? Mitosis, meiosis.
Yes, sir.
You want to revise this?
Yes.
Hmm. Okay, no problem. Monohybrid cross. Meiosis, random fertilization, 46 variations with genetic. Let me go through the book. Give me some time, please. This is it is Gregor Mendel. Then this experiment inheritance, now genotypes. This is monohybrid cross, monohybrid cross. Ways of presenting genetic information pedigrees. Codominance. Yes, this is okay. Okay. Let's get back to ancient times. Let's get back to ancient times. Ancient times. Where should we go back? We should be going back to 18th century, no, 19th century, 1880, 1865, 1800s, 19th century or 1800s. Now you are in 1800s, and what you don't know right now? You have understood everything prior to this, prior to ancient times, prior to Gregor Mendel. We have understood the genes and etc. Right now, I have told you everything, but forget it for once, for not forever, for once. Forget it. What are the genes, DNA, what are the alleles, what are the genotype, what are the phenotypes, and what are the translation and transcription, what are the proteins? Forget it for once. Let's go back to 1800s. No concept of gene, no concept of chromosomes, and no concept of genetics. If there were any concept, all those were, according to modern times, all those concepts were false. But we are right now in 1800s, and there was a priest known as Gregor Mendel. Gregor Mendel. He actually was a very sharp person, and by any chance he became a priest. But actually, he had two very great teachers who helped him to be such a great person of scientific history. Gregor John Mendel is right now a most recognized name in the field of genetics. Most recognized name. And I should tell you one thing: whenever a beginner starts an experiment on genetics, on inheritance, he or she always follows the ways of Gregor John Mendel. He is the father of genetics. But we are in 1800s, we do not know anything about it. Gregor John Mendel was a great person and he was a great student. Two of his teachers helped him to become a great man. One of them was a mathematician, and the other one of them is a botanist. Whenever I study about this scientist, I used to become emotional. Why? Because this scientist is a historical example of what a teacher can make a person. Historical example. All the research of Gregor John Mendel is based on these two subjects, and it is due to these two teachers. One was the mathematician, another was the botanist. All the research. So I used to become very emotional that a teacher can create, or a teacher can destroy a student to become a great man. So leave it. So he was a great scientific observer as well, a very intelligent student and scientific observer as well. At that time, the scientists didn't know about the of inheritance pattern of inheritance. They didn't know about how the or the children offsprings or the children sorry how the offsprings get the characters of their parents get the characters of their parents. How does it happen that offsprings get the characters of their parents? Does it happen? Keep this in mind. There is no concept of mitosis or meiosis. uh, there was a concept prevailing at that time that only mother the sorry the child is a mixture of the features of mother and father. And this is a very wrong approach because the right now we do know that a child can have the characters of his parents, grandparents, grandparents, and also their forefathers. He or she can get the gene from very upper you may say that बहुत ऊपर की जो पीढ़ी होती है जिसको हम उर्दू में वर्ड यूज करते हैं very upper generation, he or she can get the gene. So
Sir, I can't hear you. Sir?
since when you cannot hear me?
Sir, I couldn't hear you. Like, all. If there was something you said right now, I wasn't able to hear you because your voice was lagging a lot.
Okay, what did I tell you last? What did you understand at the last moment before the voice was gone?
You were telling me about that the child is a mixture of the feature of their parents, and that's a wrong
Yes.
concept.
Yeah, that is a wrong concept according to genetics because the child is not only the possessing the characters of the parents, but also the grandparents. He or she can possess the characters of grandparents and grandparents, and also from the upper generation as well. So this was a concept that was prevailing, and there were many mistaken concepts about the genetics of the offsprings, genetics of the children. So what did he observe? For example, let me tell you one of his concept that there were He started his experiments on pea plant. Gregor Mendel started his experiments on pea plants, and pea plants have two types of seeds. Sometimes they create the seeds which are yellow in color, and sometimes they create the seeds which are green in color. So, what did the Mendel do? He crossed one of the yellow parent to the parent which was possessing the green seed. Let me repeat this. The pea plant produces two types of seeds, yellow seeds and green seeds. And Mendel started the experiment. For example, this is the start, this is the beginning of the experiments of Mendel, in which he started by cross cross fertilizing or crossbreeding one of the parent, crossbreeding the yellow seed producing parent to the green seed producing parent. Assume that this is a father and this is the mother plant, father plant. When he crossed these two organisms of the same species but of different characters, one is producing yellow seed and one is producing green seed, what happened? That all the offsprings produced the yellow seeds. Does this make a sense? While you have said in 1800s, it was very much strong concept. This child is a mixture of the features of mother and father. So where is a mixture here? There is no mixture here. The yellow was crossed with the green, and when they crossed and then when they produced offsprings, all the plants which were born produced the yellow seeds. A bit of confusion is here. There is a problem. So, okay, then Mendel very intelligently started another experiment. What did he do? First of all, he mentioned that this is a parental generation or P1 generation. P1 or P generation. These are parents. So he mentioned them as P. P means parents. And this is the first generation, जिसको हम उर्दू में कहते हैं पहली पीढ़ी है ये. First generation. This is known as F1. F stands for filial, and filial means son. Son, whatever was the gender. F1. He mentioned that first generation produced by the P generation is called as F1. So what did he do? He very intelligently once again crossed, but he did not cross the parents again. He cross fertilized, cross bred the F1, जिसको आप cousin marriage कह सकते हैं. Yes, Zainab, you had raised your hand.
Sir, your voice stopped for like some seconds. Can you repeat what you were saying?
Okay, I said that the first generation which he crossed was mentioned as P by him. P means parental generation, and their immediate offsprings were called as filial generation. Immediate offsprings were mentioned as filial. Filial means son, and he labeled that immediate generation as F1 generation. F1 filial one, yani, which means this is the first generation. First generation, and this generation totally disobeyed their concepts. What were the concepts? That an offspring is a mixture of its parents. It it has misled the this concept is misleading, which I told you earlier, that the child is a mixture of the features of mother and father. So this does not appear as a mixture. So he was a bit worried at that time that if a child is a mixture of mother and father, then how come all these offsprings produce the yellow seeds and none of them produces the green seed? So this was a bit confusing. Then again, what did he start? He started another experiment, a very intelligent experiment. He designed another cross, and now what did he do? He did not cross the parents again, but he crossed the male and female plants from the filial generation, F1 generation. That is why I mentioned here F1 cross F1. Yeah, which means that the immediate generation after the parental generation, he crossed them with each other. For example, this is the male and this is the female. So yellow cross yellow, isn't it?
Yes.
When we will be crossing the F1 cross F1, so it means F1 is showing the yellow seed. So it means yellow cross yellow. Now what happened? This is another bit of confusing concept that 75% of generation showed the yellow seed, and 25% of the generation showed the green seed. Now, where would you fit this concept? Mixture of features of only mother and father. You cannot fit it here. If these are yellow, then there should be only yellow seed. And if there are green, if these are green, then it should be, if it is yellow and green, then there should be both type of seeds, or there must be some mixture of yellow and green in the first generation. So there was no mixture, and then he again fertilized, all were yellow, but they produces the green seed. So then the Mendel suddenly realized that something is getting shuffled. Remember, he does not know about the genes. Something is getting shuffled. कोई चीज ऐसी है जो कि shuffle हो रही है. It shuffled in the P1 cross, and then it shuffled in F1 cross F1. So there must be something, and something is getting shuffled. Number one was his conclusion. Number one was something is getting shuffled, and the second one was the shuffled things are in pairs. The shuffled things are in pairs. Why? How did he concluded this this point? Can you tell me? Shuffling things are in pairs. How could he conclude this point?
Because in the second experiment, he got both sets of seed, yellow and green.
Yes, 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 yes. If these were not in pairs, then there must be only one type of appearance, either yellow or green. There must be there must not be any second color appearing in this F2. For example, let me mention it here, F2 generation. This is F2 generation because this is the second generation after the first. So there, if it is not in pair, then why yellow cross yellow is producing green? Why yellow cross yellow is producing green if these are not in pairs? So he concluded that the thing that is getting shuffled, it is present in the pairs. So now what did he start, or what did he now I'm going to tell you about the written work of Mendel. Written work of Mendel. Number one, he said that yellow is dominant, dominant character, because it is appearing in 75% of population of F2. Yellow is dominant, which automatically makes the green as what?
Recessive.
Yes. So now pattern of writing. अब pattern of writing देख लेते हैं. Pattern of writing, dominant. How to write the dominant? The dominant would be mentioned as only Y, but in capital. Yellow, we would be taking the first alphabet of the dominant character, and that is yellow. And we would write it in capital form. How would we write the recessive form? The recessive form would be written by the same alphabet but in smaller form. This was all established by the Mendel. No modern scientist established this type of writing. No, this is only established by the Mendel. So uh, let me revise the pattern of writing of genetic crosses. This is the complete statement: genetic crosses. Number one, we would be taking the first alphabet of the dominant character, and as dominant, mentioning it in the capital form. And for recessive character, we would be taking the same alphabet but in smaller notation, smaller notion. So then Mendel restarted the experiment. Let's have an analysis of our previous experiment. Mendel crossed yellow with green. Yellow with green. It means that, and he also have mentioned that shuffling things are in pairs. So what did he do? Just imagine the intelligence of this scientist. What did he started? He he did write YY cross YY. This YY is mentioning the two unknown things which we actually know that these are alleles. YY are alleles and YY are also alleles. These are male, these are from males and these are from females, or you can mention female here or male here. But he said that shuffling things are in pairs. So uh, in place of yellow, he mentioned capital Y, because according to his conclusions, the yellow is dominant. And if the yellow is dominant, then the thing behind the yellow is in pairs and both are dominant. So he according to his ऑब्जर्वेशन, क्योंकि उसको ये नहीं पता था कि क्या वाकई वो पेयर्स में होते हैं या वो शफल करते हैं कि नहीं करते, ये तो एक जस्ट लाइक जैसे हम फ्लूक कहते हैं ना, तुक्का, ये उस तरह का. सो कैपिटल वाई, और फीमेल को उसने ग्रीन कलर को उसने स्मॉल वाई कर दिया. Then he said that these are shuffling. I often get very much worried that he was a gifted scientist, I think, who calculated the exact precise phenomena of modern scientists. He calculated at that time. How? There is a phenomena occurs in meiosis in which genes shuffle with
Yes.
each other. So he said that the thing behind the yellow color is in pairs and they shuffle. So उसने क्या किया? He mentioned both of these separately. He shuffled them. Same for this organism, female organism. For shuffling, he separated both capital Y, and same he did wrote for the female, separated both Y. Let me tell you that these are actually the sperms and these are eggs. Eggs and sperms are formed by the meiosis. And you know very well uh, that while the meiosis is occurring, both sets of 23 get separated from each other. And if there are, be very attentive, if there is one y in this set of 23 and there is one y in this set of homologous 23, what would happen? One y would get separated from the other y. This is the theme of this cross. One y would get separated from the other y. So he separated both y from each other. And then you know that if this is a sperm, then it is meant to fertilize with the egg. It is meant to combine with the egg. So assume that this sperm is combining with this egg and it has the allele, dominant allele, and it has the recessive allele. So these both are recombining in the next generation. So this is once again after fertilization, this is offspring F1 generation. This is F1 generation. By looking very closely, what do you conclude that the what should be the color of the seed by looking at this expression? What should be the color of the seed?
It would be yellow
Can I ask why? Why did you said this?
because Y is dominant and
And
the
yellow is
small y
dominant.
is recessive. That's right.
Yes. Okay. So does it mean that 100% yellow would be the result of the F1 generation?
No.
Uh, by looking at this observation and this observation, you will come to know that approximately there would be 100% yellow seeds because in all the organisms, in all these expressions, all the sperms are containing capital allele, dominant allele, and all the eggs are containing recessive allele. Which means if this sperm joins with the egg, none of the egg would is producing the normal protein, but all the sperms are producing the normal proteins. It means that in F1 generation, all the sperms are giving the normal gene to the next offsprings, upcoming offsprings, and normal genes is a gene is creating the yellow seed. So it means that if all the sperms are containing the alleles for the yellow seed, then all the offsprings would be yellow. All the offsprings would be yellow. Are you understanding this?
Yes.
Okay. Now, what did he perform in the experiment after F1? F1
Sorry, could
cross
you
F1.
repeat your question?
Oh, I answered it myself. My question was, what did he perform in the experiment after the F1? So he performed F1 cross F1. So once again, okay. Stop. Tell me the genotype of this expression and this expression and this expression. I am numbering it one, two, three. Tell me the genotype of this, and this.
The genotype for the first one is YY. For
Mhm. Yeah, okay.
and for the second one it's small y, and for the third one it's capital Y small y.
I mentioned a very specific pattern of genotypes: homozygous dominant, heterozygous dominant, homozygous recessive. How would you mention how would you give the this type of genotypes to expression 1, 2, and 3? What should be the genotype of expression 1?
Yeah. So for the first one, it would be a homozygous dominant as both of the Y's are capital. For the second one, it would be a homozygous recessive as both of them are small letters, they're not block letters. And for the third one, it would be a heterozygous because there's one capital Y and one small y.
Uh, very good. Mostly, what we often at the beginning of the genetics for heterozygous, we also mention heterozygous dominant, which I didn't mention while I was telling you earlier. But for the heterozygous expression, we complete it by mentioning heterozygous dominant. So this is very delightful that you have got the genotypes immediately. Idea of genotypes. It is very. I'm very happy. So F1 cross F1. Okay. Did you know about the concept of genotypes before this?
Yes, I learned it, but I was confused about it actually.
Okay. So F1 cross F1 mean capital Y, small y, capital Y, small y, right? Capital
Yes.
Y. So both have the genotype of heterozygous dominant. Now, coming to the shuffle, capital Y would be separated from the small y, and this capital Y would be separated from the small y. Now you have to cross these all these gametes because these are sperms and these are eggs. So these sperms must be fertilizing the eggs. So now, what is the point here? You now have to design the of genotypes, probability of genotypes and phenotypes. Probability of genotypes and phenotypes. How would you design a probability? This is where the statistics comes. I mentioned that one of the teachers of Mendel were mathematician and other was the botanist. Till now, we were working according to the botany. We were crossing the plants. But now, to guess the probability, here comes the statistics. Here comes the statistics. How we are going to apply the concept of statistics on this? If we have multiple features, we draw a square and we further divide this square according to the given features. For example, we are going to mention the types of sperms on the vertical side. Sperms. What are the types of sperms? How many types of sperms are there? Capital Y, small y. And we are going to mention the types of eggs on the upper horizontal. Sorry, this is vertical. Upper horizontal axis. So, how many types of egg alleles are present in the eggs? Capital Y, small y. So, to guess the probability of fertilization and probability of genotypes, you will first cross capital Y with capital Y, capital Y, capital Y. What should be the genotype of this expression?
Oh, sorry, what? Could you repeat it, sir?
What should be the genotype of this expression?
It should be a homozygous dominant.
Okay. And what should be the phenotype of this expression?
Phenotype. Um, it would be a yellow. It would be a yellow plant.
Yes. Well done. So now let's multiply this capital Y to this small y, capital Y, small y. It has a genotype of heterozygous, but what should be the phenotype?
It would be yellow because it has a dominant allele in it.
Well done. So now coming to the lower y, we are crossing this small y with capital Y. So capital Y, small y, it would have the heterozygous dominant genotype and yellow phenotype, same as this one. Now coming to the last cross, probability is this sperm would be fertile. If this sperm fertilizes this egg, what would happen? Small y, small y. This, tell me the genotype and phenotype.
The genotype for this would be, yeah, it would be homozygous recessive, and the phenotype for
Hmm.
this would be a green plant.
Green seed.
Oh,
Okay. So it means that right now our probability says, our probability is probability ratio. It is actually called as probability ratio. That yellow, and there are two types of probability ratios. One is phenotypic ratio, phenotypic probability, and the other one is genotypic probability. Okay, if I uh, if I mention it in the form of ratio, out of 100, sorry, let me mention it properly. Let me make a proper table. Phenotype, how many types of phenotypes we have? We have one phenotype as yellow and the other phenotype as green. So according to this square, this is called a Punnett square. It is called a Punnett square. Uh, if I assume that we have 100 plants, what is the phenotypic probability of yellow and what is the phenotypic probability of green?
at least, I think it would be 75 by 100.
75 by 100 or and 25 by 100. And if we mention it in the form of ratio, we can mention 75 ratio, sorry, we can mention 3 to 1 ratio. Yellow to green ratio is 3 to 1 ratio. Isn't it? Understood?
Yes, sir.
Okay, so it means that we have come to know the probability that the phenotypic ratio of this cross would be 3 to 1. The dominant character would be taking the part of the population, major part of the population, 3 by 4, and the recessive would be 1 by 4. Coming to the genotypic probability, how would we design the genotypic probability? For example, how many types of genotypes do we have here? One is homozygous dominant, ratio, heterozygous dominant, and homozygous recessive. So if I ask you that if we write the genotypes in the form of ratio, what should be the ratio of all these three genotypes out of four?
For homozygous dominant, it should be 1. Ratio 2, ratio 1.
Well done. This is the genotypic probability of the next generation. And guess what? Guess what? Look at this expression. What did we mention? That after F1 cross F1, Mendel got 75% of the dominant character. This is phenotype. This is phenotype, not genotype. And this is 25% of the recessive character. Coming back to the written work, 75% of the dominant character and 25% of the recessive character. This is the same probability in the written work as in the practical work. But guess what? What Mendel hypothesized that something is shuffling. The shuffling things is in pair and yellow is dominant is 100% correct. 100% correct. This is the most um, amazing thing that sometimes shakes me, that how intelligent he was. He just hypothesized that something is happening here, something is shuffling, and this is in pairs, and blah blah blah. And after that, he came to the very same conclusion he drawn a few days ago, because the pea plant takes some time to produce offsprings. Few days it takes. So this totally eliminates the concept that the offsprings are mixture of their parents. Actually, you know what actually Mendel did? Mendel actually showed us what are the patterns of inheritance. How does the inheritance work? How the genes shuffle? This is Mendel showed. Okay, coming to the a more technical word. In your curriculum, this is the last thing. I know the time is past, but this is the last thing I'm mentioning here. Yes, predict probabilities of outcomes from monohybrid cross. This is a term that we have. If you read this curriculum, then you come to know that Mendel is not mentioned here. But this is a word that demands that Mendel should be mentioned, or Mendel should be taught, because without Mendel you cannot understand a monohybrid cross. Without Mendel you cannot understand. So let me clear this concept. Monohybrid cross. Monohybrid. Monohybrid means when F1 is heterozygous for a single allele. Single allele. Uh, is this F1? Is this heterozygous or homozygous?
Heterozygous.
Yes, and it is heterozygous for a single allele. So when F1 is heterozygous for a single allele, it is called as monohybrid cross. Monohybrid cross. This is a monohybrid cross which we have just done here. And what was I going to mention? The monohybrid cross. Yes, there are some other, for example, uh, there are some other types of crosses. For example, if I mention y capital Y small y capital T small t. So now this F1, assume that this is F1, but there are two heterozygotes, two heterozygous combination. One is this and one is this. This type of cross would be called as dihybrids. When the F1 is heterozygous for two alleles. When it is heterozygous for single allele, it would be a monohybrid cross. Okay, now just tell me one thing. What did you improve? Because you said that you know about you have studied it already, and you knew about a bit about the genotypes. But what did you improve in this lecture?
I was able to understand the Punnett square in a better way and what it actually is, not just mugging up the concepts just like that, but actually understanding them.
So yes, you were going to say something.
And I also understood genotype in a better way.
So the revision is giving you benefit, right?
Yes.
This is the phenotype, right? Phenotypes of the parents, and then after that they cross it, and this is the Punnett square, and after that this is result 3:1 ratio is here, and this is genotypic ratio mentioned here. Okay, you can. I was just showing you the book. You do not need this table, this whole table. You do not need to understand because it is not mentioned there. Actually, you already needed to describe the monohybrid cross and the probabilities, how to design the probabilities. Yes, fine. Okay, anything you want to share?
No, sir.